Someone needs to mute their computer. I'm getting all kinds of wild sounds coming through. Ah, much better. Okay. So this is part of the pre-lab. Um, okay, so this is going in your lab notebook now. You need to have the title, purpose, um, structures in terms of the chemicals we're using. Like today and Wednesday we're using where the lab is on alcohols. So you want to have those structures that we're going to be using uh, down, and you can use this as a reference. And along with the structure, have the first aid. Remember, the first aid can be had from the SDS document, section 4. So for example, you would go on this one here, you'd go to Google and you do a search. And you're going to end up with a PDF file. Everyone, you need to mute your computers, please. Okay, so you do a search on that and you get a number of hits and look for a PDF. And it'll be the SDS, and it's the first aid is section 4, and it's standardized internationally. Okay, so you're going to need structures plus the first aid. We're having challenges today. Materials.
Anita, can you please mute?
A, B, C, D, E. Thank you, Anita. Then you're going to need a procedure. And that's the part that completes the pre-lab. Now, sometimes there are, I'm going to back up here. Sometimes there are pre-lab questions, not all the time. Sometimes there are. If there are some, that needs to go into the pre-lab as well. Now, this is your first wet lab. So anything that isn't done properly, you can fix it on this first lab. And this first lab is lab 5 on alcohols. Next week, we're doing lab 4 on orange steam distillation. Okay, to finish the lab write-up, you're going to need a data table. And the data table is simply a place to put your results. Anytime you have to calculate something, anytime you observe something, anytime you are measuring something, that's what goes in the data table. And then lastly, every lab needs a conclusion. And that's something very simple, you know, a paragraph that just summarizes what you've learned. That's all it is. Not a big deal at all. And you always have 1 week to do the lab. Okay, so did that answer your questions?
Yes. So whatever experiment we do in class, that's gonna go on the lab manual, not the write-up. Write-up would be just the conclusion, the data table, and as the safety for the chemicals.
No, nothing goes in your lab packet. Everything goes in your lab.
Notebook?
Yes, everything.
Oh, okay. And that'll be due at the end of the week, not on the same day.
It'll be due a week. I always do it on, I always have a double section. And I always do a week from the day that the second lab is. So in other words, it'll be a week from Wednesday. It'll be due. So Monday people have 9 days to do it. Wednesday people have 7 days to do it. And once you've done the pre-lab, there's not a lot to do to finish up the lab.
Professor, I have a question.
Yeah.
Just to clarify for the lab notebook, there's like you just want a composition notebook. It doesn't matter if it's crafted or not, right?
Correct.
Okay, perfect.
Correct.
Thank you.
Now, some students up in Sierra were finding pretty good deals over at Walmart on lab notebooks, and they were finding really nice ones for like $3. Um, I mean, so if you know, as opposed to, I think Tammy said she got hers for 50 cents. Is that true?
Yes, 50 cents at Walmart.
Yeah. So
I think they're 47 cents now.
interesting trend since everything else is going up, this is going down.
Snuggle.
Yeah. So cheap ones are just as good as the really nice ones. I'm just saying though that that one for $3 is a real good deal. I think the pages are pre-numbered and everything. It's so if you got a spare three $3, which is what it costs for a cup of coffee at Starbucks, a cheap cup of coffee at Starbucks. That's either the American one or the just plain old drip. So anyway, just FYI. Okay, so any more questions about the um, the pre-lab write-up and what lab we're doing? Uh, Now you're going to be also today and Wednesday, we're going to have to check in our drawers. All we're going to do is I'm going to assign a drawer to you and your partner. And sometimes you work in twos, sometimes you work in fours. Like next week, we're going to be working in four groups of four. This week, going to be groups of two. So you and your partner will share a drawer. So I need to, and then there's going to be a combination inside. And then I'm going to open it with I have a key that can open up the back of the combination lock. So I can get you into the drawer so you can start your lab. And then before you leave, I want you to make sure you can do the combination. Now, if all of you have had 305, it's exactly the same deal as 305. Same locks, same kind of combination as before. Most schools use the same kind of locks. All the schools I worked up in Washington all use the same locks. Um, so they're pretty universal. High school uses them, middle school uses them. They're just all over. Okay, so any more questions on the lab write-up? And then this is a different version of it. These are some examples. By the way, we always have two signatures. One's for cleanup, one's for pre-lab. So you can peruse those when you get a chance. And these are in modules. So this is the process. You do this at home, pre-lab. While you're in lab, you do the lab, fill in the data table, answer the questions, do a conclusion. Every single lab. Dry labs, you don't have to do pre-lab at all. You just fill it out, turn it in, upload it. Now you're going to be scanning all the pages in your lab write-up for that specific lab. So lab 5, you're going to take a picture of the pre-lab part, the data tables, the questions, and the conclusion into one PDF file. I'm going to be uploading that whole thing under assignments. Okay. All right, so let's jump over to.
Sorry, so you had posted for alcohols and phenols a report sheet. So you're saying we're not filling out the report sheet, it should be filled out on our lab write-up?
Yes. Yeah.
Okay.
Everything goes into your lab book. Now,
Okay.
like in 305, a lot of the teachers just had you fill in some questions, and that was it. This is a formal write-up. Now, when you take any other chemistry classes in the department, Chem 400, 401, 420, whatever, they use this form that I'm talking about here, where you handwrite everything into a lab notebook. So when I took over this job a couple years ago, teaching 306, that's one of the things they made sure that I did, is to have the 306 students do a complete lab write-up. Okay, are we feeling okay now with this lab write-up? And again, remember, you can correct anything you miss on this first lab. So I wanted to alleviate the pressure because you never had a wet lab from me before. Okay, well, let's jump back over to aldehydes and ketones. Let's finish this up today and then get into carbohydrates a little bit. Now, I want to do a quick review on these acetals and ketals again. This is the trickiest part of this aldehydes and ketones chapter. Okay. So whenever we have an aldehyde or ketone and an alcohol, it forms a new additional product where the two are combined into a new product. And then it continues on to yet another product that's combined. So as I was stressing last time, we always want to start with our backbone, the primary backbone. Okay, and that starts with either the aldehyde or the ketone. The added alcohol is going to form the secondary backbone, which attaches to the anomeric carbon. Now, this anomeric carbon is going to come into play big time when we talk about carbohydrates. That is going to be the focal point of where carbohydrates attach to one another, and also where the reactions occur in carbohydrates is that anomeric carbon. So it's crucial you understand the concept of anomeric carbon, because it's a backbone of all the chemistry in carbohydrate chemistry and aldehyde ketones, this anomeric carbon. Okay, now last time I gave you a little flow chart to figure out whether you have an acetal, ketal, or a hemi version of that by answering these three questions. Is there a carbon that is surrounded by, or a single bonded oxygen, two oxygens to a single carbon? Yes or no? If yes, then you have something. Okay, and that again is the identification of the anomeric carbon. So if yes, then you have an anomeric carbon, and yes, you have an acetal, ketal, hemiacetal, or hemiketal. The next question is, regarding the anomeric carbon, is there an H present? If there is if this is yes, then you have a hemiacetal or an acetal. If there is no H present on the anomeric carbon, then you have a ketal or hemiketal. And lastly, also attached to the anomeric carbon, do you have a hydroxyl group present attached to that? If yes, then you have a hemiacetal or a hemiketal. So this is important little process to go through whenever you're trying to figure out what's going on. Now we're going to use the same deal, same little process here when we talk about carbohydrates, because we're going to be looking at the anomeric carbon in a carbohydrate and whether it's a acetal or hemiacetal. Now, carbohydrates mostly deal with acetals, not too much with ketals, except for fructose and fructose related things. And the identification of hemiacetal is particularly important in carbohydrates because that'll help us identify whether the carbohydrate is a reducing sugar or not. So this process is real important. That's why I started. Okay. So let's apply it to this guy here. Okay. We arrived at this by taking aldehyde. In this case, it's ethanol or acetaldehyde and methanol. Okay. So it's a one carbon alcohol added to a 2-carbon aldehyde. So you have to establish the first backbone, and then you establish the secondary backbone, which is going to be the alcohols. So all the action is on the anomeric carbon. Everything attaches to the anomeric carbon. So the alcohol is going to attach to the anomeric carbon first, and that's going to give you the hemi. And then it's going to happen again to form the full-blown acetal. Now, in carbohydrates, it depends on whether you have another alcohol present or not. Now, remember, before we talk about carbohydrates, I want you to keep in the back of your mind, though, carbohydrates are polyols. They have a ton of hydroxyl groups on them. That means they act like alcohols. So that's where so if you have a aldehyde around a sugar, it's going to use the alcohol part of that sugar to form the hemiacetal. And I'll show you in big time detail how all that happens. So this is a really important part of this aldehyde ketone chapter. So the hemi part is the OH part. No OH, no hemi. And again, it all centers around the anomeric carbon. Okay, all right, so let's do some examples here. So this is butanal or carbons, and this would be ethanol, 2 carbons. So this is kind of what we're doing here now. Okay, when we have this would be the aldehyde, this is the alcohol. Okay, so identify the primary backbone, which is the aldehyde or ketone. And this is going to be the secondary backbone. And this is going to be the anomeric carbon. So this is going to attached there. Notice I'm doing a double arrow because these are reversible reactions. Okay, so what we end up with is here's the anomeric carbon, and then we have the alcohol. So here we have OCO. That means we have a anomeric carbon, and we have OH present, and we have H present. So number one, we go yes. Second one, do we have an H present? Yes. Do we have an OH present? Yes. So this is the OH, this is H, and this is OCO. Okay, so when we have an H present, that means it's an acetal or a hemiacetal. And OH means it's a hemi. So that's a hemiacetal. Okay, so let's now it would stop here if there wasn't another OH present. And we're going to find with carbohydrates, there is no additional OH present because this is formed in a ring structure. So as far as carbs go, it would stop at the hemiacetal stage. However, if you're not dealing in carbs, what happens is the additional alcohol will then go ahead and react again. So we've got so we have our yellow primary backbone, anomeric carbon. We have our two alcohols. So we've got our full-blown ketal. So OCO, yes, we have OCO. That means we have an anomeric carbon. Yes on H, there's an H right here. And no OH. So, excuse me, not a ketal, that is an acetal. So the fact that there's an H there means that it's an acetal or hemi acetal. And because there's no H, it means it's a acetal. So that is a typical reaction. Okay, are there any questions about this little test or whether identifying what it is? Okay, all right. Um, and then if you start with a ketone rather than an aldehyde, you're going to end up with this, which is where the H would be. Instead of an H, we have a carbon. Hence, it's going to be a ketal because it came from a ketone. Now, some books don't generalize. I mean, they generalize this to everything is an acetal. And other books differentiate between the starting point of whether it's a ketone or an aldehyde. I like to differentiate between the two in this class so that you know what you start with, whether it's an aldehyde or ketone. In a more advanced class, they just call everything acetals, and you have to figure it out on your own. Okay. Now, sometimes you can have on the same molecule both an alcohol and an aldehyde. And as long as the molecule is 6 members, well, actually 5 members or more, the tail end can come around and react with the front end. So think of like a scorpion. If the scorpion's tail is long enough, it could bite itself. Same kind of deal with this. So here we have the alcohol part. This is the aldehyde part. And this is over here, OCO, anomeric carbon. We have H present. That means it's an acetal or a hemiacetal. OH present, that means it's a hemi. So this forms a hemiacetal. Now that hemiacetal, if there's a free alcohol around, it can form a full-blown acetal. Now in carbohydrates, this is an adjacent carb that has a whole bunch of OH groups available. So when you string glucoses together, those are all acetals, except for the end one, which is a hemiacetal. And I'll get into extreme detail with that. Okay, now these acetals or ketals can come apart in an acidic environment with water and reform the individual aldehydes and alcohols. And again, this can happen biochemically as well via enzymes. Okay, so this is the additional part of Unit 2 for our flow chart. This part on the end. So I have a combined flow chart that's got all the stuff up above into one chart. Now these are some common aldehydes and ketones. Formaldehyde is used a lot in the formation of various kinds of epoxies and glues, and that's what is in the materials they build houses with, unfortunately. So brand new houses have a poison floating around the air inside of a brand new house, and it's called formaldehyde. They're even stamped on it. If your house is built in California, it's built on it, stamped on the, it's called OCB, I believe, oriented. Does anyone know what OSB stands for when you're talking about sheet goods for building? The O is oriented. Anyway, it's used in place of plywood. It sort of looks like plywood, but it's made from chipped wood rather than sheets of thin wood. Anyway, The other thing is it's used to preserve specimens. So whenever you're in an anatomy physiology class and you're dissecting something, that funny smell is what you're smelling is formaldehyde mixed with water. And it does a really good job on preserving tissue. Now, the problem is if you're inhaling formalin into your lungs, and that's the reason why you smell it, is because you're inhaling it into your lungs. It's also doing a good job on preserving the tissue inside your nose, inside your sinus cavity, and inside your lungs. So it's not good to be a anatomy physiology teacher because you're surrounded by this stuff all the time. Students get a little bit of it for a while and then it's fine. But if you teach this where you are constantly around formaldehyde, it's not good. Of course, in chemistry, we have our own brand of not good stuff too, but particularly with anatomy physiology. Okay, most common ketone is acetone, and you see it everywhere. You see it in nail polish remover. You use it around epoxy as a solvent. It's all over the place. And biochemically, aldehydes and ketones are very present. They're all over the place. These are a couple hormones, but it's just all over the place. Aldehydes, particularly, are very fragrant smelling. And that's what we're going to be using for lab 4 when we do the steam distillation of orange peel. We're going to be looking at a chemical similar to that. Now we're going to be using that. You're going to save it, and you're going to use it to put in your soap you make it smell citrusy. A lot of the artificial flavorings are aldehyde based. Now, you do not have to know all these, by the way. These are just FYIs. Okay, so that's it for aldehydes and ketones. In the next test, you can have 3x5 cards, by the way. And all the rest of the tests, you can have 3x5 note cards for the test. So are there any questions about this guy? Aldehydes and ketones. Now I have additional worksheets. And with answers in Canvas files, Unit 2 worksheets. Reactions, practice. Okay, so are we all ready to start carbs? I can tell you are. Okay, so you have all heard of carbs for a long time in your life. They perform a bunch of functions in our body. And we're going to go into detail in Unit 5 on that guy there. You're going to go in detail on how glucose specifically undergoes oxidation to give us lots of adenosine triphosphate energy molecules plus carbon dioxide and water. So a biomolecule is a general term. Biochemistry is a study of that biomolecule. Okay, so always remember this part. Carbohydrates are polyhydroxyaldehydes and ketones. So they include not 100%, but mostly an aldehyde or ketone and lots of hydroxyl groups or alcohols. Sometimes they call sweeteners polyols, and that is does not contain an aldehyde or ketone. It's just hydroxyl groups. You've all heard of this sugar ribose, as in DNA, RNA. Now, the one thing this this set of slides doesn't do much on is oligosaccharides. So there are 4 classes of carbs. Monosaccharide, disaccharide, oligosaccharides, and polysaccharides. And it's just how long is the chain? Now, this monosaccharide is a single glucose or fructose or galactose unit. You're going to see in a minute here that the most common monosaccharides are 6 carbon units, and they form 2 kinds of rings, a 6-membered ring and a 5-membered ring. The other thing is when we get into biochemistry, we're going to find in this unit and the unit on lipids that we have something called enantiomers. And enantiomers are mirror images of each other, or stereoisomers. And they're, excuse me, and they're designated with the D or L designation, kind of like cis or trans. Well, in this case, it's D or L. Now, this is extremely important in biochemistry. And the reason why it's important is because enzymes, active sites, are designed specifically for a D or an L version of the substrate. Now, there was a huge, devastating problem back in the early 60s in Europe and the United States. There was an anti-nausea medication called thalidomide. Thalidomide is an anti-nauseous drug given to first trimester pregnant women to prevent nausea. And it exists as both the D and the L form. Unfortunately, one of them was anti-nausea, and the other form caused birth defects, major birth defects. It's such a tragedy. These children were born without arms or legs. It was horrible. And it had to do with this D and L form. So the drug manufacturer did not purify it beyond a certain point that distinguished between the D and L forms. Really a tragic, tragic occurrence. And it all had to do with stereoisomers. Now, there's something in organic chemistry called chiral carbons. Chiral carbons have 4 different groups attached to it. Doesn't matter how big the group is, as long as it's different. So in this case here, we've got a aldehyde, a hydrogen, a hydroxyl group, and a carbon chain. 4 different groups that therefore this exists as a D form and an L form, depending on what direction that goes, to the left or to the right. Now you can have more than one chiral carbon in a molecule. Now, this was a question on an MCAT test I was helping a student with a long time ago. And it specifically asked about chiral carbons. I don't know if it's still there or not, but I know the version I was helping it with did. So let's look at a very common monosaccharide. So again, it's 6 carbons, 1, 2, 3, 4, 5, 6. And in this case, it's not in a ring, it's open. So we're going to find 5 and 6 membered carbon, let me back up, 5 carbon and 6 carbon monosaccharides can be open or closed down into a ring. Most of the time, 90% of it, well, probably even 95% of the time, the ring version is more stable than the open version. Hence you always see glucose and fructose, all of them in the ring formation. That's because they're more stable, but they do open up. Every single one of them opens and closes. Now, the deal is when they open, of course, they expose the aldehyde, which of course is prone to reactions. Now let's look at these, this guy here. Okay, so carbon number 1 at the top here, it only has 3 groups attached, so it cannot be chiral. It has to have 4. Carbon number 2, it's got 1, 2, 3, 4 groups that are different, so carbon number 2 is chiral. Same with number 3, same with number 4, same with number 5. Carbon number 6 down here has 4 groups attached, but 2 of them are hydrogens. So this is carbon number 6 right here. Hence, it's not chiral. So this particular monosaccharide has 4 chiral carbons. So this will definitely have a D&L version. Now there's a formula for figuring this out. I don't want you to worry about that. The main thing is the concept of if there's a chiral carbon available, you have D&L forms. Now there's different ways to draw carbs. We can do it ball and stick, which we never do. And we do something called a Fisher projection. Which isn't very common also. But we use it to illustrate the different forms of D&L. So if this is the Fisher version or drawing, we put the aldehyde part at the top. And then as we go down, the OHs are going to go to the left or the right. The one farthest from the aldehyde, farthest, determines whether this particularly monosaccharide is either D or L. Okay, if it's to the right think of dex as in right-handed, and an L as in left hand, or going to the left. Now, again, this is not the form that we're going to be using much of in here. Another interesting thing about carbs is they will actually rotate the light if they're a D or L version. They'll usually go to the left or the right. And the degree by which they turn the light, the polarized light, is a function of its concentration. So the more it turns, the more concentrated that sugar solution is. When I did undergraduate research a couple of different times, and one of the times I worked for a carbohydrate chemist, and we used this device in the lab quite a bit. Okay, okay, that's the main thing I want you to know on that. Excuse me. Now, I did mention that the lipid did I mention lipids were virals, DNL? I forgot what I said. Proteins are anyway. Amino acids are active. Carbohydrates have monosaccharides and amino acids are both chiral. The most common monosaccharides are D, and with amino acids, they are L. So, now, like in our naming conventions, we've got mono, tri, tetra, penta, hexa. We're mostly going to be working with pentoses and hexoses. And there's two versions of each one of those. Depending on whether it is a keto version or an aldehyde version. And this is more common, the aldehyde version. So when we're working with hexoses like fructose and glucose and galactose, there are 16 versions for each one of those. And a specific name for all those. Now we're only going to learn 3 of the 16. And we're not going to do L's, we're only going to do D's. So there's 8. So we're doing 3 of the 8. Glucose, fructose, galactose, the most common of them all. Okay, so aldotriose, aldehyde, 1, 2, 3, triose. Aldehyde, 1, 2, 3, 4. So tetrose. Now, these are the 8D 6-membered monosaccharides, and so they're hexoses, and because when they open up, there's an aldehyde, and This is what glucose looks like with the Fisher projection, and here's galactose. Now, fructose is a ketohexose. So it does not have an aldehyd in it, it has a ketone in it. And that one we're going to learn as well. Now, everyone knows carbs. There's a reason why carbs are an addiction, is because they are sweet. And sweetness kicks off dopamines in your brain, and you are in la la land while you're eating something sweet. My neighbor requested next time I go to Rayleigh's is to pick her up some heavy duty sweet cupcakes. She says she's been craving them. Now she's about to die, so it doesn't really matter if she has all this sweet stuff or not. But she craved it, so I said, "Oh, no problem. I'll pick you up some next time I'm over there." Notice there's a reason why fructose is used a lot instead of glucose. Look how much sweeter fructose is than glucose. The reference is sucrose, and sucrose contains fructose and glucose. We'll see in a minute. So they set that as a reference point of 1. And then everything is either sweeter or less sweet than sucrose. But fructose is almost twice as sweet as sucrose. Now, an invert sugar is a mixture of, no, let me back up. Sucrose contains 2 monosaccharides, glucose and fructose. An invert sugar, like honey, they're not connected, the glucose and fructose. They're just present as separate glucose, separate fructose. So invert sugar is more sweeter. It's sweeter than sucrose because of the fructose piece is separate. And then fructose by itself is just sweeter than heck. So when you have corn starch and you release some of the glucose, commercially, you enzymatically convert the glucose to fructose. This was invented in Japan. And once you start releasing all these fructose monomers from the long starch, it gets sweeter and sweeter and sweeter. It's a very simple process also. And additionally, it's really bad for you. Okay. So basically, anything that has 5 carbons or more usually exists as the cyclic hemiacetals or ketals. Now we're going to be using the Hayworth structures to draw all these. So we always put the anomeric carbon on the right. The anomeric carbon is carbon number 1. In the case of fructose, it's carbon number 2. I'll show you that in a minute. Now this happens to be galactose. And we're not going to use the D because we're always going to use Ds. So you may include D in your naming of these monosaccharides or not. But they exist as an alpha and beta version, however. And that depends on what direction the anomeric OH, the OH part of the anomeric carbon is located, up or down. If it's up, it's the beta version. If it's down, it's the alpha version. Now, the way the carbon's in a 6-membered ring, fructose is a 5-membered ring. 6-membered ring, now this is a heterocyclic ring because it doesn't, all the units are not carbon. There's an oxygen in there. So it starts, the anomeric carbon on the 6-membered ring is going to be carbon number 1 and goes around and then up to the carbon that's above the glucose ring. This is going to be beta D ribose because the anomeric carbon's hydroxyl group is going up. Now, if we draw it, normally it's drawn like this, line drawing wise, instead of like this with the CH2OH. Now, this is a big deal, cyclization of glucose and fructose and galactose, actually. So we start with I'm going to end on this too. This is kind of wildly complex. Okay, so let's look at, now because the OH group here is going up, it's a beta glucose. Now you're going to have to learn how to draw these. You don't have to include the hydrogens, but you do have to include the OH groups. So the way I remember to draw glucose is I go up, down, up, down. So for me, glucose is the easiest to remember because it just alternates up, down, up, down, up, down. Now I'm talking about where the OH groups are, not the hydrogens, just the OH groups. And I always use the beta version because that's the one that goes up. Anomeric carbon, I just put a dot by it. So do we have an OCO here? Yes, we do. We have OCO. So this is going to be a acetal, hemiacetal, hemiketal, or a ketal, one of those four. I'm going to use our tests. So yes, OCO. Yes, hydrogen's present. Yes, an OH is present. Hence, this is a hemiacetal. Oops, this is OH. So this part here is the hemiacetal part. Now, remember in our last chapter on aldehydes ketones, if there's another alcohol around, that hemiacetal will then go to a full-blown acetal. Well, guess what? Here we have another glucose molecule, and this end hydroxyl group is functioning as an alcohol in terms of this acetal reaction. So this is going to form the secondary backbone. This is going to form the primary backbone. So then this alcohol now is going to attach there. So what you end up with is, let's draw this differently. So this is glucose, and this is alpha glucose actually. And this can either be alpha or beta over here. Let's just do it the same way. Let's do alpha glucose over here as well. Okay. So we have anomeric carbon right here. We have no H's. That means it's a ketal or acetal. And no OH. So this ends up being then a full-blown acetal here. Now, that's on the left-hand glucose. On the right-hand glucose, we have OCO again. We have H, and we have OH. So that's a hemiacetal. Now, it's really important to get practice at identifying when you're looking at a carb, whether it's what kind of carb is it? Does it contain only acetals, only ketals? Does it have a hemiacetal present somewhere? Because if it's got a hemiacetal present, it is called a reducing sugar. And we're going to talk a lot about reducing sugars in this chapter. Okay, so I'm going to leave it here because you covered a lot of stuff here. And carbohydrate chemistry is aldehydes and ketones on steroids. Not because it's harder, it's the structures are more complex. That's what makes it harder. So really, we're just isolating out the anomeric carbon chemistry each time. And there's a particular name, how these are linked together. This is called a glycosylic linkage, and you have to name it, and you name it by, this is carbon 4, this is carbon 1, and that hydroxyl group's going down, so this is the alpha 1 or glycosylic linkage. And we're talking about that on Wednesday. Okay. Chemistry of carbs, pretty wild stuff. Excuse me. Any questions about distinguishing and also using this? I use this gimmick that I invented a long time ago a lot to determine whether we're looking at a hemiacetal or an acetal or whatever. Okay. So if there are any questions, I'm going to end it here. And what would be really good practice between now and Wednesday would be starting looking at those worksheets I showed you and making sure the aldehyde ketone chemistry is solid. Because you can see this is all aldehyde and ketone chemistry. And with a lot of extra baggage slapped on it with the structures. Okay. Okay, so there's no more questions, I guess.
Professor, I just have a quick question.
Sure.
I was wondering, you know how for Unit 1 you had the reaction posted and you said that chart is going to get longer and longer?
Yeah.
I couldn't find it on Canvas.
Well, then let's look for it then. This is I don't know what this should be Unit 3. This is dealing with acids, carboxylic acids and esters. I don't know why it's over there. Okay, there's this one, which is a zoomed in version of the bigger one, with a little more detail and then structures that indicate like this is hemiacetal, this is a full acetal. So this is handy, this version, but I should have, let me do this here. This is a hard drive in my laptop. Yeah, this is the version that you were talking about, right? Okay, so I'm going to put that here. So go to Unit 2 worksheets. Thank you for asking that.
Thank you, Professor. I know you have the zoomed in version, but I think it just helps me understand better when I can see where it actually comes down from.
That's why I created them. Yeah, I agree.
Yeah, thank you.
Yeah, because some books don't do that. They don't show because when you're trying to figure out reactions, like you're trying to synthesize something, that flowchart really helps a lot. Because you can say, well, what are the precursors to get to here? You just follow the arrows backwards. And that's how to get there, So to me, visually, it makes a lot of sense. And that's why I created it. So, okay, any other questions? Well, thank you for asking me that because I didn't know it was not here. So,
No, thank you.
okay, so I'm going to go ahead and end this and create the file and video file and everything to give myself some time for it to do its thing. And then I'll upload it and everything. And I'll see you in class. This alcohol lab is a pretty good lab. It's going to be your first wet lab. Next week is going to be your steam distillation. It's not a hard lab, but it's complex. The apparatus is pretty wild looking. It's got all this expensive ground glass joints and everything. And then you've got to take the orange apart. Oh, by the way, everyone needs to bring oranges next week because you're going to be taking the zest from the orange part of the orange, not the white pith part, only the orange part. And then you're going to be boiling it and then distilling off some of the essence in there. So it's pretty cool. Then we're going to save that and then put it in our soap that we make. That's a cool lab too. Okay, so any more questions? And if not, I will end this. Okay. All right, everyone, we'll see you in a little while. Oh, I'm bringing the corrected test with me as well. So if you're in the Wednesday lab, and if you're on campus, drop by the Monday lab. I can give you your test. Otherwise, I'll pass them out on Wednesday. Okay, I'm going to end it now. Take care, you guys.